So thank you very much for coming along. This is my presentation entitled "These Boots Are Made for Walking, Free Living Gait." So as an overview, I'm gonna tell you why walking, why gait is important for us. Then I'm gonna touch base on the VTIME study in which myself and Lisa have been involved. And I'm gonna focus on the results that we have achieved. So I'm gonna tell you a little bit more about what we have learned. So why walking? Why gait? Walking is important because it's a very sensitive biomarker. So people are using biomarker quite often in these days. But what's a biomarker? A biomarker is simply a quantity that we can measure and tell us something about your health status, how you're doing. It's like when you go to the doctor and you measure your blood pressure, for example, or when you measure your temperature with a thermometer. It tells us something about how you're doing. For example, if you have high blood pressure or high temperature, it might tell us that something going wrong. The same thing with gait. Gait is like having a biomarker of your health status and telling us, for example, if you're getting older, so if you're aging, we're getting slower, or for example, if someone has a pathology, we can detect some information by just measuring how you walk. But you think walking is quite easy. It's not easy at all. Walking is a complex task, and especially safe walking. It involves movement, so it involves moving your limbs, your legs. It involves keeping your balance. But also something that we never think of is it involves the brain. So for example, while walking, you might be carrying an object or a cup of tea, so you have to pay attention on what you're doing. It also involves control. For example, you might be talking to someone, you have to take decisions. You have to, for example, cross obstacles. So it's a very sort of intense and load for your brain. And if something goes wrong in one of those three domains, so either the brain, the movement, or the balance, you might experience a fall. So it's not so unusual to experience a fall because, as you can see, almost like a third of people aged more than 65 years fall each year. And it's a very interesting topic, and for this, the World Health Organization has also written a report about how to avoid fall and how to manage falls. So it's a very hot topic. So now I'm going to touch base on the VTIME study. So the VTIME study, as you can see, is a virtual reality treadmill combined intervention for enhancing mobility and reducing falls in the elderly. So basically it's an intervention study which aims at enhancing mobility, so make you move better and reduce the number of falls. As you can see, this is a multi-center project, and it has involved 5 different centers: Nijmegen, Leuven, Tel Aviv, Genoa, and Newcastle. In total, we have recruited 300 participants across the 5 different centers. We have recruited elderly fallers, people with mild cognitive impairments, so with some attentional, for example, and memory problems, and people with Parkinson's disease. All those people had to have at least 2 falls during the 6 months prior to be part of the study. So what I'm really going to focus today is the data that we have collected in Newcastle, and I'm going to really talking about enhancing mobility, so trying to make people move better. So I'm going to talk about the movement bit of the equation that we've seen before. So just a brief introduction to the study. So we ask our participants to come along at baseline before intervention. And then each participant underwent an intervention consisting of 3 40-minute sessions per week for a total of 6 weeks, for a total of 18 sessions. It was a randomized intervention, so some of the participants were randomized to a treadmill training, so they were asked to walk on a treadmill, and some of the participants were asked to have a treadmill training plus virtual reality intervention, which, as you can see, consisted of a television and a camera. Each patient wore like 2 markers on their feet, so the marker could be detected by the camera, and the feet were displayed in the television in front of them, so they can have a feedback about the position of their feet. They were asked to complete some tasks, so for example walking through a park, crossing obstacles, crossing hurdles and puddles. and they constantly had feedback about the position and of their feet. After the intervention, we asked participants to come back 1 week post-intervention, 1 month, and 6 months post-intervention to assess again their gait, their mobility, and their cognitive status. So what have we asked our participants to do during testing? We asked a lot. So for each of the assessments, so baseline, 1 week, 1 month, and 6 months, we asked them to perform some walking tests, balance tests, to complete some questionnaires, color trails, to complete something about how they felt about falling, so fear of falling, falls efficacy scale. We asked some questions about how active they were, and also how, in general, their health was. And at the end, we also perform a computers-based assessment. So we asked them a lot to do. But today we're going to really focus on the walking test, so the gait test that we perform at each assessment point. So what we asked was to come along in the lab and to walk at a normal pace on a 10 meters walkway. As you can see here in the video, we asked them to walk back and forward at their normal pace. While doing that, participants were wearing a little sensor that you can see here on the lower back. And our sensor is like our thermometer, as I said before. It's our tool to measure gait. After the laboratory testing, we also ask our participants to wear the sensor for 7 days continuously in their real-world environment. So at home, it might be in a shopping center, or it might be also outside. Why did we ask them? Because really, if we think about that, the testing in the laboratory is not a complete real picture of what really walking is, because of the equipment that we ask participants to wear, because the floor is so flat, there's no obstacle. Although the patient told me, "Oh, Silvia, this is not like my real walking, because there's nothing here, it's just so smooth." And then a lot of wives said, for example, "Oh, I've never seen my husband walking like that." So it's obvious that there's something going on, when people are testing in the lab, and they always are behaving their best. So to really get a realistic and a real picture of the way we walk during our everyday life, we ask them to wear the sensor, as I say, for 7 days at home. So when people are really tested in the laboratory, what can we evaluate? So the little sensors, like the motor wrist device to, for example, count the number of steps or your activity, is like having a magnified glass that can really magnify the signal that we are leaving while walking. So acceleration, we can't see it, but we can really measure it with our little sensor. And using the sensor, we can really measure and evaluate a lot of different gait outcomes, measures, and characteristics that, as you can see here, can be divided into different domains. For example, pace, rhythm, variability, asymmetry, and postural control. This is a model that has been validated by Dr. Silvia in 2013, so that we can really see that those variables are sensitive, so are really important to describe walking, both in aging and also in pathology. But let me take a step back and really, what is a step? So while you're walking, if you think of, you're leaving some footprints on the floor, a step is simply an event between two foot or heel contact. And once we can detect the step, we can also derive a few measures. For example, step length, so how long is your step? For example, 50 centimeters. How long it takes to take a step, which is the step time, for example, 0.6 seconds, and obviously how quickly you can take your steps, so step velocity, for example, 0.8 meters per second. So we can get a few information once we have detected steps. And another important concept is step asymmetry. Obviously, when we are walking, our right side is not behaving as our left side. So for example, as you can see here on your left, we have a symmetric pattern. What does it mean? It means that our right and our left step behaves the same. So our right side and left side behave the same. Whilst, for example, on your right side, you can see an asymmetric gait. And as you can see, the right side is different from the left side. Now, obviously, we would like to be as symmetric as possible, but as we know, everyone has a sort of degree of asymmetry embedded in their self. So really what we want to do is to reduce asymmetry as much as possible and to try to make the right side behave as the left side. And then, when we ask people to wear the sensor in free living conditions, so when they are at home, for example, we can derive, different measures. But as you can see, for example, on the top, you have an example of a signal collected while people are walking outdoor. Or on the bottom here, we can also see, for example, that people are doing different things when they are at home. They're sitting down, they're walking, they might be shuffling around the kitchen, they might be walking downstairs, lying, walking while talking. So there's a different set of activities. What we really did to analyze the data in free living condition, we did measure just the walking bouts, so when someone was walking, so we detected the walking activities, and then once we detected those, we can really tell how much time, for example, you spend walking per day, so for example, an hour or two per day, the total number of steps that you can take, and the mean bout length, so on average, how long you walk for when you are walking. What have we found? So what are the preliminary results? So those are just preliminary results about related to the VTEM study. So as you can see here, I'm just presenting a total of 52 participants divided by 18 elderly followers, 6 people with mild cognitive impairment, and 28 people with Parkinson's disease. Those are the results that we have really collected in the lab. So you will see in the different graphs that in blue are the results before the intervention, in red the results after 1 week of intervention, and then in green the results that we collected after 1 month post-intervention. And as you can see here, step time, if we focus on people with Parkinson's, so PD, we see that step time decrease, so you're sort of taking a step quicker. If you look at step length, we can see that actually step length increase, so you're taking longer step, which is very good. And in fact, if you look at step velocity, so how quick you are, step velocity increase after intervention. So this is very good because it means that you are walking, with more confidence, longer step, quicker step, and walking faster. So very, very good. If you look again at the asymmetry, do you remember, we would like to be as symmetric as possible. If we look at step time asymmetry, so the difference between the right and left steps, after intervention, asymmetry decreases. So you are walking with more symmetric gait pattern. So again, a very positive outcome. And if you look at the result when you are at home, in your free living environment, your sort of everyday life, we saw that walking time per day increased up to 2.5 hours per day. We can see that number of steps increased as well. And also the mean bout length increased. So it means that you're walking more and you're walking longer. So again, positive results. So the take-home messages, I think, are, two. After the intervention, you walked better and in a faster way in the lab. And also you walked more when you were at home. So overall, our participants benefit from the intervention. I want to acknowledge all the sponsors and also thank you very much for listening.